My name is Tobias Buchborn. As already announced, I'm a psychologist with a PhD in neurobiology, and I'm currently working with the Institute for Psychopharmacology and the Central Institute of Mental Health. I'm a researcher, and all my research so far has been devoted to psychedelics. And when speaking about psychedelics, wait a minute. Sorry. When speaking about psychedelics, we deal with the drug. And in order to understand what the drug does and how it actually ends up changing the psyche, we need to understand certain other parameters as well. So before we can actually go to understand what the drug does to the psyche, we beforehand have to understand what the drug actually does to the brain. And before we can actually enter the brain, we need to ask the question, how does the drug actually go from the mouth all the way up to the brain? How does it find its way? How does it find its way out of there again? And also, once it is there, what does it actually do there? And only in this triangle of psychology and neurobiology and the pharmacological basis, we are actually able to fully understand what psychedelics are about to do. The two main pillars of pharmacology are pharmacokinetics and pharmacodynamics. These provide us with all kinds of parameters that help us to understand the journey of a drug, including psychedelics, through the body. Here is the agenda for today. We will focus on serotonergic hallucinogens, which I consider synonymous with the term psychedelic, so LSD-like drugs. And we have basically three columns we will be talking about. We will talk about pharmacokinetics and the processes that are relevant for pharmacokinetics. We will speak about pharmacodynamics. And in the end, it's basically about diving into current literature and seeing what are hot topics at the moment when dealing with psychedelics and how can we look at these topics from a pharmacological perspective. When dealing with literature, it's always important to be critical, to read the paper carefully, not right away buy into what is said, but instead trying to ask questions. I think asking questions is the most important thing when dealing with science in general and when dealing with literature. Pharmacokinetics. So I said pharmacology has two columns. We got the pharmacokinetics and the pharmacodynamics. And the pharmacokinetic asks the question, what does the body do to the drug? So once it is intaken, what does the body do it? Where does it bring it to? What does it do to the molecule? How does it get rid of it? So it's basically about the journey of the drug through the body. This is what pharmacokinetics means. Kinetics means movement, and pharmacon is the drug. In pharmacokinetics, there are four main processes, which are often abbreviated as ADME. A stands for absorption. This process deals with the question, if I consume a drug orally, or I put it under the tongue, or I put it transdermally or rectally, how does it actually find its way into the bloodstream? Because only from the bloodstream it is actually able to end up in the brain or wherever you want to have the drug. So this is considered absorption. How does the drug find its way into the bloodstream? Once it is in the bloodstream, we deal with distribution, and pharmacokinetics knows different models of how to describe them. The easiest one is the two-compartment model, which assumes that there is a compartment where a lot of blood is, so the vessels themselves, but also highly perfused tissues. And there's another compartment, which is the peripheral organs, which are less perfused by blood, such as muscles, fat, and skin. And distribution asks, once the drug is basically in the blood, how does it find its way to the organs? Once it is distributed, but also already on the way to its distribution, a drug also comes across the liver. Some enzymes can also be found in the gastrointestinal tract and all kinds of other organs as well. And within the liver, the body tries basically to get rid of the drug. It would cut off pieces from the drug or add certain groups to the drug so that it becomes easier for the body to get rid of the drug. And the idea is also to inactivate the drug by working enzymatically on it. So this is what metabolism means. And the last step is excretion. This is the question once the body has traveled, once the drug has traveled our body again and again and again in hundreds of circles, how does the drug actually finds its way out of the body? And here, of course, kidney and the feces are the most important ways. And we will now go through these and discuss them with regard to LSD. This is the psychedelic I chose because we have most literature about LSD. When dealing with the pharmacokinetics of a drug such as LSD, we don't always have all the information for a certain drug because understanding how one drug, it's a very small molecule, actually finds its way through the body is not so easy to understand. What can help, however, is if we know a little bit about the properties of the molecule. We know that LSD is a relatively small molecule. Here you see the molecular weight, which means it's more easy for LSD to travel the body because it can basically slip through the cracks, so to speak. We can also look at the log P value, which is around 3, which is a very good value. It basically means if you ask LSD, left is a room full of fat and right is a room full of water. Where do you want to go, LSD? LSD would rather prefer to go into the fat room. Some molecules would also find their ways into water, but LSD is more lipophilic. We also know that LSD is actually a base. It's not an acid, although we sometimes refer to LSD as acid. It's a base. It's a weak base. And this means when it is in an environment where there's a lot of acid, such as in the stomach, it's actually protonated. And these values already can tell us something about the journey of LSD through the body. But let's go through it step by step, how this actually works. When we assume that we consume LSD orally, for example, by swallowing a blotter, then we can assume that LSD would travel down the esophagus and would eventually end up in the stomach. Some people might assume that LSD now is being absorbed from the stomach, but this is rather not the case because of its pKa value within the stomach. LSD molecules would be protonated, which means they accept an H plus proton, and this makes LSD charged. And when LSD is charged, it actually cannot travel out of the stomach because it cannot pass the membranes. That's why LSD is most likely transported into the small intestines. Here the pH is much lower, and here LSD molecules are less protonated. So here it's actually much easier for LSD to go through the endothelial cells of the gastrointestinal tract. It's most likely, given its high lipophilicity, that it travels through the membranes to actually get to the other side, but we cannot rule out that it also slips through the cells, through the endothelial cells, because of its relatively small size. Okay, once it actually left the intestines, it goes via the portal vein into the liver, which can be seen up here. And then it's basically carried along with the blood to the heart. The blood is then carried further to the lungs because the blood is meant to be added with oxygen. Then the drug, along with the blood, will go back to the heart. And here it's being pumped, so to the heart, to the lungs, and back to the heart. And from here, it is basically pumped. So from the liver to the heart, to the lungs, back to the heart, and then it's actually pumped into the system. And this pumping into the system works via the aorta, which can be found up here. And the aorta actually has multiple exits. So some of these exits would travel down the body, some of them would go back to the body, and some of them would actually go back, would go all the way up to the brain. And before the aorta actually enters the brain, it would actually emanate into the carotid artery. This then branches in front of the brain, and part of it would go to the face and the other part would go into the brain. And actually, Um, After the drug has been absorbed, which is down here, as I mentioned before, we already speak about distribution. So as soon as the drug is being pumped into the system, we speak about distribution. This is what I told you before. The drug would be carried via the internal carotid artery to the brain. And here the carotid artery would go multiple branches, such as the anterior cerebral artery, which carries the LSD to the frontal cortex. And this is one of the main regions where we believe that LSD is active. Now, we need to have in mind that LSD is transported via the blood. So for now, even if it's up here in the anterior cerebral artery, it is still within the blood. And the drug still needs to enter the interstitium. This is where the neurons are. So it actually needs to leave the blood to find its way into the synaptic cleft. And this is not so easy actually, because there is something that is called the blood-brain barrier. And here you actually see how the blood-brain barrier works. Here you see a brain in a sagittal section. If we enlarge this little area, we can see it like that. This region, which I'm highlighting now, is basically the vessel. And here the blood flows, you see all kinds of blood cells. And within this blood flow, you would also find billions of LSD molecules. And these actually now need to go across these endothelial cells. And usually it's not so easy for a drug actually to cross the blood-brain barrier, which is composed of astrocytes. These are these ones here. And they are also composed of these proteins, which create very tight junctions between the endothelial cells. These proteins, which tie them very closely together, are called claudin and occludin, which already tell you that they are meant to really make sure that the endothelial cells are very close and no drug can pass. But the thing with LSD is, I mentioned it before, LSD doesn't need to go through these cracks because it's highly lipophilic. So it's actually quite easy for LSD to travel across the membrane of these endothelial cells into the interstitium where the neurons are. So this is true for all the psychedelics. Psilocin would also be able to do it, and so forth. Serotonin, for example, is not lipophilic, it's rather hydrophilic. So serotonin would not be able to do the trick. It would not be able to enter or go across the blood-brain barrier. LSD, however, is able to do it. And once it is here in this interstitium, it is where it belongs, namely in the synaptic cleft, where it induces its pharmacological effects. Yeah, we will speak about the pharmacological effects later, the pharmacodynamic effects, but of course, we also need to ask the question, still speaking about pharmacokinetics, once LSD is within the brain, how is it actually gotten rid of? And this also works via vessels, but this time not via arteries. So arteries bring LSD to the blood, but it's the veins that carry LSD out of the blood. Here you see in blue these veins, and they have all kinds of branches here into the cortex. The smaller branches are called venules. This is where LSD would basically find its way through the membranes into the veins, and then being transported to these bigger veins, which you see up here. This is the so-called superior sagittal sinus, and actually it transverses both hemispheres. It's a very massive vessel. Here, all of the LSD is being carried to, and then it would be transported further across other sinuses, would go down to the internal jugular vein, and then would again find its way to the heart, to the lungs, to the heart, and then via the systematic circulation go to the liver and the kidneys. It's important to know that a lot of LSD molecules actually can travel the body hundreds of times. So it's a matter of time that more and more LSD molecules would be metabolized in the liver and then being gotten rid of by the kidney or by other organs. A minor part, so apart from the removal of the drug by the veins, a minor part can also be might also be removed by the lymphatic system or by the lymphatic system, and certain proteins such as aquaporin 4 might facilitate the exit of drugs by the glymphatic system. But it is assumed that the major pathway of LSD leaving the brain is probably via the sinuses. Okay. When there, of course, this was now basically explained on what we know about pharmacokinetics in general, but of course, people also want to do more specific research on the drug itself, and this was done in the early days by radioactively labeling LSD and then injecting it into the cat or into the rat, and then you just need to visualize the radioactivity, and then you can see at which time point does LSD accumulate in which areas. And what they actually found in the cat, that it would highly accumulate in the bile. This already tells us something about the excretion. We will talk about this in a bit. We will find a lot in the plasma, of course, compartment number 1, we spoke about it. And medium levels in the lungs, the liver, the kidney, and the brain. So these are all areas which are well perfused. LSD always goes with the blood, and less LSD we would find in the intestines, in the spleen, in the heart, and the muscle, and the fat. So this is rather the peripheral compartment where it's more difficult actually for blood to find its way to. Similar things have also been shown in the rat, where radioactively labeled LSD was given to the rat, injected, and it would also highly accumulate in liver, kidney, lungs, less in spleen, heart, and brain. So these are, again, the well-perfused areas, and only little of it in fat or in muscles. So this is actually well in line with what we generally know about the distribution of drugs that, mainly whose destinies mainly are determined by the blood flow, basically. In humans, so these were animal studies. In human studies, what people often do is such a plasma curve, which is very interesting. So you would give LSD to your patients, and then you would basically take the plasma at several time points, and then you can basically see at which, so this is the absorption phase, basically. So how long does it take for LSD to be absorbed? And this would then be the elimination phase. And here you can determine parameters such as the Cmax. This is basically the maximal concentration you find in the blood after application of the drug. You see it's here in this range. You can also determine the time point when the peak is. And this is very often correlated with the peak of the effects, which makes sense. The more drug is in the plasma, the more likely it is that it also finds its way into the brain then. You can also determine the half-life of the drug, and then you can, if the drug follows first-order kinetics, basically predict how much it will take until the drug is cleared from the system. With first-order kinetics is usually that with every half-life, which is 3.6 hours, 50% of the drug within the body are being gotten rid of. So if you at the beginning have 4 nanograms, after 1 hour you have 2 nanograms, after after one half-life you have 2 nanogram, after another half-life you would have 1 nanogram, and so forth. So this is what first order kinetics means. So this is something that helps you predict the course of the drug effect. And also when you have drugs where you want to repeatedly apply something, a plasma curve can give you an idea when it's best to reapply the drug, so to speak. We have now spoken about absorption. We have spoken about distribution. The next step we want to talk about is the metabolism. A very important parameter when it comes to the metabolism is actually bioavailability, or F. Basically, the question is, if you consume LSD by a mouse, for example, how much of it would actually find its way into the system, or how much is actually destroyed by the liver in the first round. And we know for LSD that the bioavailability is relatively high, so we can actually say that most of the LSD we consume also finds its way into the system, and only little is being metabolized in the first round. So it needs multiple rounds for LSD to be metabolized. In pharmacology, we differentiate phase one metabolism and phase two metabolism. For LSD, we know there are about, I don't know, 20 or 30 different metabolites. So the pharmacology of this drug is very complex. So I only chose a few examples for you. Here you got the original LSD molecule. One thing the liver does in phase one is basically add a keto and a hydroxy group. This is so via hydroxylation and oxidation, this is meant to increase the hydrophilicity of the drug, so it's more easy to get rid of it in the urine. This is the idea behind it. It's also meant basically, if this is the active molecule, the liver wants basically to change the molecule by adding these groups, so it's not active anymore. So this is the evolutionary idea behind it. Then there is a phase two. So once you, for example, this is another metabolite, it's called 13th or 14th hydroxy LSD. So this is one metabolite, this is another one. After this phase one has taken place, you would basically add another thing to the drug, which is a glucuronic acid, which is being attached to this hydroxy group, and then you enter with an LSD glucuronide. And this is also meant to make the drug more hydrophilic, so you can get rid of it easily. So these are examples of how the body actually deals with LSD to get rid of it while it travels via distribution the body again and again and again. And if we have a look at the plasma curve, then you can estimate actually how long it takes for liver, kidney, and bile to get rid of LSD. Something that's very interesting to mention in the context of metabolism as well is psychedelic prodrugs. You probably have heard of them. Psilocybin, for example, psychedelic and magic mushrooms is such a prodrug. As you probably know, psilocybin itself is not able to pass the blood-brain barrier. This is due to the fact that it has a very low log P value. This means it is not really lipophilic. In order to get into the brain, you need to be lipophilic. And psilocybin is then dephosphorylated to psilocin, and this drug has a higher lipophilicity, and that's why psilocin is then actually able to enter the brain and induce the psychedelic effect. Importantly, so this is called a prodrug. Psilocybin is the prodrug for psilocin, basically. Importantly, the fact that psilocybin is a prodrug does not necessarily mean it's not active. It is indeed not able to go into the brain, but it actually binds to a lot of receptors, which also can be found in the peripheral system. And as to this, it's possible that psilocybin in the periphery has effects. This is not very well studied, but chances are that psilocybin is not totally inactive, at least not in the peripheral system. Other examples for prodrugs are these new LSD versions, which you probably have heard of as well, are currently being synthesized to somehow circumvent the law. LSD is illegal, so that's why a group is attached to it. And once this new drug is then made illegal, then another group is attached to it, and this is like a cat and mouse game. And the idea is here that these groups, this 1P group, 1B, and 1V group, and it goes on, is basically cleaved by the liver to liberate LSD. And thereby you would consume it. They are all by now all illegal, but they used to be legal. So the idea is here, I consume a legal drug, and then within my body, the illegal drug is being synthesized, but then you don't have a problem. So this is basically the idea. Then there's actually within the field of tryptamines and phenethylamines, there are really a lot of prodrugs, but another one would be psilocybin. I think it was synthesized by Nickel, I'm not completely sure. And this drug is also thought to be a precursor to psilocin. So speaking about metabolism, prodrugs are important to have in mind. LSD, however, I thought not to be a prodrug, but to be active itself. Okay, how is LSD excreted? This has also been investigated in humans. A lot of the research has been done in Switzerland, actually, and it was found that if we consume LSD orally, only 1% of this LSD would find its way into the urine. And 60% of the LSD consumed would find its way into the urine by this metabolite we've already talked about. This tells us two things. Number one, either LSD is massively metabolized and or LSD and its metabolites might be excreted via another pathway as well. I personally believe it's a little bit in between. In these human studies, they very often only investigate LSD and one or two metabolites, but we know that there are much more. So it's possible that other metabolites can also be found in the urine. But we know from rat research, again with the radioactively labeled LSD, that a lot of the radioligand is actually recovered within the bile and only 5% in the urine. And the bile actually leads the drug into the feces. This is basically what you see here on the right. We got the liver. Here you have basically the bile as it is generated in the liver. This is being collected here in the gallbladder, and then it would be carried down into the intestines. And here it can basically be eliminated by adding it to the feces. And this is very often done with LSD glucuronides, and this has also been reconfirmed in this rat study. So although this kind of research is still lacking in humans, so most of the human research always focuses on the urine, it's not unlikely that a lot of the LSD and or its metabolites can actually be recovered in the feces more than in the urine. But this will probably need more research to have a clearer picture on that. Yeah, so far we've spoken about the pharmacokinetics. As I said, pharmacokinetics is about the question, it's basically about the journey. How does a drug find its way to the brain and back again and out of the body? And pharmacodynamics is not so much interested in these questions. It rather asks, once the drug is at its target site, so in the brain, for example, what does it do there and how does it do it? Dynamics means something like force, so it's basically the force or the effect of what the pharmacon does, what the drug does to the body is basically the question of pharmacodynamics. And as I understand, most of you who joined today are students of psychology, so probably you have heard or you have seen the synaptic cleft already. Again, we would enlarge a part of the brain very strongly and would find here a neuron and here a neuron. So this is basically the postsynapse and this is the presynapse. Here an action potential would arrive and would make the vesicles basically become one with the presynaptic membrane, and then the content is being released to activate the postsynaptic receptors. This can be ion channels, but it can also be G protein coupled receptors. And LSD would basically we spoke about the blood-brain barrier, so LSD would come out of the vessel into the interstitium and would find its way here into the synaptic cleft and bind the receptors. We can actually think of LSD as speaking multiple languages. As you might know, there are different transmitters within the brain, and transmitters like dopamine, noradrenaline, and serotonin, glutamate, GABA, and so forth can indeed be understood as different languages. So some neurons speak these languages, others would understand them. And LSD, when we stay in this picture, would be speaking multiple languages. The reason for this is here you see the LSD molecule that it actually shares important constituents of the neurotransmitters. So it shares the indole nucleus with serotonin, and it also shares the benzene ring. This is this one with dopamine and noradrenaline. In addition, they all have their amide chains, so this makes them actually amines. And this is the reason why the brain misconceptualizes LSD as being dopamine or serotonin or noradrenaline. This is how one can imagine it. So LSD would basically prefer receptors that are somewhere within these systems. And this has something to do with the structure similarity. A very important parameter when speaking about drug receptor interaction is the affinity. It's a really, really important parameter. And you will find in the literature two measures of it, KD and KI. They actually mean the same, so you can treat them alike, but they are determined in a different way. KD is a measure of affinity and KI is a measure of affinity. The difference between both is that KD can only be determined if the drug you are using is available as a radioligand. So if some company has produced LSD and attached some radioligand to it, then you can determine the KD. Very often, however, this is not the case. And this is why you have to determine the affinity indirectly by using, for example, radioactively labeled ketanserin and measuring how well LSD is in displacing ketanserin binding to the serotonin 2A receptor, for example. So this is the difference between both. But let's focus at the KD. This is actually quite easy to understand how this is measured. You have a system, you have a brain system, or you have a you have some cells you're working with in your lab, and you would basically increase the concentration of LSD more and more and more and more. And the more you do it, the more the drug would be able to bind to all of the receptors within your system, may it be the serotonin 2A receptor. So at some point, you have so many LSD molecules in the system that all of the receptors are occupied. This is then saturation. And by convention, the concentration of a drug which occupies half of all receptors is the affinity. And you would basically see, okay, this is where is the concentration? In this case, it would be around, I don't know, two nanomolar, let's say it's 2 nanomolar. So you would basically have to go to the 50% binding and then look what the concentration is. So this is what affinity means. Affinity is actually the likelihoods that your drug interacts with a given receptor at a given concentration. And this can be done with all kinds of receptors. Here, for example, done by Lichty, a team from Switzerland. We got serotonin receptors, we got noradrenergic receptors, dopamine receptors, and also some transporters. And here you basically see the affinities of LSD for all of these receptors. When it comes to Ki values, it means the lower the value, the higher the affinity. And based on what we heard here, what this basically tells you, if you have a concentration of LSD of 4 nanomolar in the synaptic cleft, 50% of the serotonin 2A receptors are occupied. This is what it means. And these are very important values. We will also come back to them later when we speak about microdosing and the question whether it's actually possible that at such a small dose LSD is able to interact with the receptor. This can actually predict it based on such data. So as I said on the last slide, LSD speaks multiple languages, so it can actually bind to serotonergic, adrenergic, and dopaminergic neurons, receptors. What I usually tell my students is when you look for affinity values, don't just go for one paper. The reason for this is very often a lot of papers determine one and the same phenomenon. So here, for example, if you go to the KI database, which is open access, you can go there yourself. You just need to enter the drug you're interested in, and then you will get such a list. And here is where the affinities are. All of these are all different studies, and they all determine the affinity of LSD for the serotonin 2A receptor. And as you can see, there is actually quite a drastic variation across the study. So it's actually always recommendable to go there and use a median instead of just going for one paper. Also, when you go into this database, you find all kinds of other receptors. As I showed you on the slide before here from the Lichty group, they investigated a certain amount of receptors, but of course there are much more receptors. And here in the KI database, you actually learn that LSD actually interacts with almost all serotonin receptors, and with most of them with quite high affinity. Only the serotonin 3 family not. So it's really primarily serotonergic compound, And that's why we also say it's a serotonergic hallucinogen. But also seeing this, you actually get to see that LSD has quite a lot of effects, and it's actually quite difficult to predict what all of these receptors do, especially, for example, if you chronically take LSD. It's very difficult to predict how they adapt, how they're sensitized, and so forth. The more complex the pharmacodynamics of a drug, the more difficult it's actually to predict something like this. I've mentioned it before, and I guess you also know it. The key receptor for all serotonergic hallucinogens is the serotonin 2A receptor. So if you consume your LSD, this receptor is being occupied, let's say 50 or 60%, and then you have your unusual effects as Alice here in this case. If you pre-treat Alice with ketanserin, and then Alice would also consume LSD, then Alice will be very sad because it realizes that LSD is not active anymore. And this actually has been shown by some groups in Switzerland again and again, also for other psychedelics. And based on this, but also a lot of animal research, we know actually we need the serotonin 2A receptor. This is the primary pharmacodynamic of serotonergic hallucinogens, and the other receptors might flavor the psychedelic experience, but they are not the main driver. Okay, and let's speak a little bit more about drug receptor interaction. Similar as in pharmacokinetics, there are models that describe how a drug would interact with its receptor. And an easy model is the so-called two-state model, in which we believe that when we go into the brain and we think about the serotonin 2A receptor, let's say we are in the frontal cortex. And these are all serotonin 2A receptors. Some of them are inactive in their inactive state. These are these blue, and some of them are actually in their active state. And they can all spontaneously switch. This is the assumption. But when there's no ligand around, the inactive state would predominate. So that means that there's always a basal serotonin 2A activity, even if there's no ligand present. This is the idea. If now LSD comes in, it would preferentially bind the active state, would stabilize it, and so more and more of the receptors would actually become activated. And this would then, compared to the basal output, increase the outputs. And if LSD were a full agonist, then it would basically induce a full effect, but psychedelics are usually not considered full agonists. So the idea is an agonist would basically prefer binding to active states, would stabilize them, and by the time more and more of the receptors would find their way into such an active conformation. This is the two-state model. And the two-state model is relatively simple, but it also helps us to define certain ligand types. So if LSD binds to the receptor, there are certain things it could be there. It could be a full agonist, which means it activates the receptor to the fullest. It could be a partial agonist, which means it only partially activates it. It could be an antagonist, which means it does not really change the basal activity. And it could be an inverse agonist, which means it would reduce the basal activity. We know, however, for psychedelics that they are partial agonists. So this is what is most important when speaking about psychedelics. Serotonin itself is a full agonist. So the endogenous ligand is a full agonist. Serotonin 2A receptor, as all receptors in the brain, I drink something, sorry. It's actually a protein, and a protein is a chain of amino acids. And here you see this chain. The serotonin 2A receptor is composed of 471 amino acids, and each of these letters is basically an amino acid. And this is then, being crouched together basically, and then it forms such a three-dimensional structure. And it's a G protein coupled receptor, which means it binds to G proteins, as you can see here. It's also called a 7-trans membrane receptor because this amino acid snake basically would travel the membrane 7 times. That's why we call it 7-membrane receptor. So if LSD binds, it would then initiate the downstream events. This is the activation of the phospholipase C. This then cuts off the heads of certain lipids. These heads would then travel down into the cell, into the neuron, would release calcium from the endoplasmic reticulum, and this calcium then can activate calcium-binding proteins and induce all kinds of follow-up effects. There's also another pathway here via, so I said, PLC would cut off the head of the lipids. Of course, something remains here, and this remainder would then basically interact with the protein kinase C, and this then also can induce downstream events. And what is then done really depends on the environment and the overall state of the brain. For example, what could happen is that the neuron gets aroused by this cascade. It could also be that downstream of it, some genes would be expressed so that new proteins are being generated and inserted into the membrane. Another alternative would be that the pseudo architecture of the cell changes and so forth. So this kind of LSD binds to the receptor would then go on and transduce the message into the cell. So this is the interaction that occurs here. And the question, and this refers to what I said before, is it a full or is it a partial agonist? And this can actually be decided based upon how much of this IP3 is being generated or how much of this calcium is being released. This is then an index of how full or partial an agonist is. And again, this can be measured, otherwise we wouldn't know it. And understanding such a measurement is also not so difficult. And in the context of how good a drug can actually activate such downstream event, two additional parameters are important: the potency and the efficacy. So we've already gotten to know affinity, which is the binding likelihood, and potency and efficacy refer to this IP3 accumulation. To which degree does it happen, and at which concentration of the drug does it happen? Here you see examples of 3 drugs. We got serotonin, LSD, and mescaline. Again, we have in our lab, our little our brain tissue or our cells. We would increase the LSD concentration or mescaline concentration more and more and more. And the more the concentration is, the higher the IP3 production. As you can see, serotonin is actually able to induce a maximum effect, so maximum IP3 production. LSD would only induce about, I don't know, 20% of the maximal effect, and mescaline is a little bit better. And this tells us what I mentioned before, that psychedelics are partial agonists, so they are not able to activate this signal cascade as much as serotonin is able to do this. And the potency can actually also be seen here. So you again look at 50%, go all the way to your sigmoid curve, and then go down to the concentration. And this concentration here, let's say it's 0.08 or something. This is then the potency of serotonin to induce this IP production. And this, of course, can also be determined for the other drugs. And as you can see, the potency might be higher. Here, the potency is higher, but the efficacy is lower. And this, of course, has also been done across a lot of studies. Here you see EC50 values of certain drugs and also the efficacy values. As you can see here, LSD is highly potent, which is in line with its affinity. Again, the smaller the value, the more potent the drug is, because you only need a little bit of molecules to actually induce your effects. For mescaline, on the other hand, you need a lot of molecules. So you really need tons of mescaline before it actually activates the serotonin 2A receptor. And this we also know because, I don't know, what was it, 500 milligrams or something of mescaline you need to consume for you to an effect. For LSD, you only need 100 microgram. So this plays a role here. And what I told you about the maximal effect here can also be seen here. So serotonin is usually around 1 or 100, whatever you want to call it. And then mescaline induces only 60% of the maximal response. And LSD is quite weak, actually. It's only a partial agonist and induces only 40% of the maximal response. So this is what potency and efficacy means. Once the drug has bound to the receptor, how much does it change the physiology of the downstream of the cell, and how much molecules do you need for this to happen, potency and efficacy. Yeah, as I mentioned before, if we go by the study I just mentioned here, this is a study from Newton et al. If we go by these values, we can actually say that LSD is about 5,000 times more potent than mescaline. This, however, does not mean that when you consume LSD, that the psychedelic state is more intense or something like this. Mescaline can be massively intense. It just means you need way less, 5,000 times less LSD molecules in the brain for having a psychedelic effect than with mescaline. So this is what potency means. So sometimes you probably hear in the media, oh, LSD is the most potent psychedelic. And this basically refers to this idea you don't need a lot of molecules to have your effect. Yeah, things are usually more complex than simple models. So we spoke about the two-state model. This two-state model has already been reversed. We by now assume that there are actually multiple pre-existent states. Some say that each drug induces a very own conformation of the receptor, and depending on how the drug changes the conformation of the receptor, all kinds of different cascades can be recruited. So it's not only about IP3, which I showed you before. Where is it? Yeah, somewhere around here, I guess. Phospholipase C. It's not only about this, but also a lot of other things are being activated. And the thing is, and this is what is called functional selectivity, there are certain psychedelics which prefer this pathway. And then there are other which prefer this pathway. And there are again other which prefer this pathway. And this is called functional selectivity. And as we will later learn, some people assume that this concept is very important to understand why certain serotonin 2A agonists are actually not psychedelic. This is a paradox we will speak about later. Also, there is the possibility of the serotonin 2A receptor to actually form bigger complexes with other receptors, and this then allows the psychedelic to recruit cascades which are actually not part of the serotonin 2A receptor, but of the other receptor. So as you can see, downstream of the receptor, all kinds of complex things can happen, but having the concept in mind of affinity, potency, efficacy, and functional selectivity will already give you some tools to understand this kind of interaction. Functional selectivity has all kinds of implications. This is actually results from my own research back in the days at the University of Magdeburg. I asked the question, why is it actually that when you repeatedly consume LSD, let's say for 1, 2, 3 days, that at the end of these 3 days, LSD is next to inactive. This is called tolerance. Whereas when you do the same with DMT, there is no tolerance. This is unusual because they are both acting via the same mechanism. And our research indeed points to the direction that the answer for this is that they recruit different cascades downstream of the serotonin 2A receptor. What we, for example, see is that LSD, but also the psychedelic drug DOI, which also induces tolerance, induce very strong internalization of the receptor. What does this mean? Here you see a cell. What you see here in red is the membrane of the cell. If you now apply DOI, you see that a lot of, so it's not the membrane, it's the serotonin 2A receptors that are sitting at the membrane. This is what you see here in red, labeled by an antibody. And if you apply a DOI, then a lot of the receptors are internalized. As you can see, there's a very thick cluster in the middle. It takes about half an hour to have such a cluster. If you do this with LSD, you also find very strong internalization, but DMT actually fails to induce significant internalization. And internalization is a very important process that the more often it occurs, it at some point ends in the receptor being degraded. So it's being destroyed. And it seems that LSD is more quick to initiate this cascade, whereas DMT is not able. And this might be a reason why LSD is strongly associated with tolerance, whereas DMT is not. The question is now, why is this the case? And what we investigated in addition to the internalization is a certain enzyme, which is called phospholipase D. So it's an alternative pathway. And I didn't include the results here, but what our results show that DOI and LSD are actually able to strongly recruit the phospholipase D, but DMT fails. And this enzyme is actually very important to induce the invagination here of the membrane. You see, in order for this receptor to be internalized here, all the way down here, the membrane needs to be invaginated, and this is being done by this enzyme. So speaking about functional selectivity, whether a drug prefers the phospholipase C or the phospholipase D can have quite drastic consequences for phenomena such as tolerance. So there's indeed functional implications of these different events that occur downstream of the serotonin 2A receptor. Okay, the last bit I would like to tell you about pharmacodynamics. Of course, beyond the mere molecular level, we also need to ask, okay, once all of these things go on in the neuron, in the individual neuron, what happens actually to the brain as a whole? And here we actually enter the field of neurobiology. Of course, focusing today on pharmacology, I can only briefly talk about neurobiology, but let's give it a go for 1 or 2 slides, I think. We know that the serotonin 2A receptor is across the whole body. You find it in a lot of regions of the body, but the highest expression is indeed here in the cortex. In the human, you would find it a lot in the frontal cortex, but also here in the visual area. We know from in vitro electrophysiology that if you apply a psychedelic into this region here, the pyramidal cells, which are localized there, the layer 5 pyramidal cells, they increase in excitability. So if you repeatedly stimulate this slice preparation, then normally these cells would habituate, but when LSD is in the game, they don't habituate. So there's an increased excitability and a reduced refractoriness. If we then move further away from these in vitro studies and go into EEG and MEG findings in humans, we know that when a lot of such cells fire together, we have these EEG waves. And here you have 3 different neurons. You see, they all fire in the same pattern. This is called a desynchronization. So the brown one would be the upper one, this would be this one, and this would be this one. All neurons fire in the same way. And if you have hundreds and thousands of this firing in the same way, we would see this in the EEG. If you now apply a psychedelic, what happens is that they totally fall out of sync, which means neurons all of a sudden start to do things that are not entrained with the others anymore. And this is called desynchronization. And this is something that really has been highly replicated across studies. So this is really something psychedelics seem to do. When you go into individual areas of the cortex, they desynchronize the activity there. If you go beyond local areas, so just beyond local, this would be local. But if you now think of networks, like a network between this and this region and this region, then we normally know from the brain that there's a certain amount of modularity. So there are certain networks, such as the default mode network, which fire and wire together. And then there's another network, which is called the salience network, which was also fire together, but they would usually not interact. If we now give a psychedelic, this modularity is basically eliminated or reduced. As you can see here, it's reduced, and these networks would less interact within the module, but all of a sudden start interacting with one another. And this is then interpreted, for example, in the context of ego dissolution. So this is just a very brief idea. All of this is also part of pharmacodynamics. So it's not just about the molecular level, but also these higher level effects. Also, actually, the psychology and the psychological effects, ego dissolution, can also be considered a pharmacodynamic effect on the level of the psyche. This is so we are actually also interested in this kind of research. So at the Institute for Psychopharmacology at the SETI in Mannheim, we also follow up on this kind of research, and we do this in animals. And the problem with EEG in humans is that EEG only gives you an overall output of the cortex as a whole, but you cannot really target the cells you are actually interested in. We are only interested in the serotonin 2A receptors that are we are only interested in the pyramidal cells that express the serotonin 2A receptor, and this is something you can do in animals. You can also, in animals, go beyond the cortex and see what's going on subcortically, what EEG cannot do. And we can also measure with high resolution transmitter release, for example, glutamate release. All of these things are impossible in humans, and this is the advantage of the methodology we are using, which is called fiber photometry. So we would basically label here, this is basically the cortex of a rat. You see, it's a coronal section of the frontal cortex. And this is here the olfactory nucleus, the piriform cortex, and this is the frontal cortex. And this band, which you see here, this is serotonin 2A receptor expression. And when we have a lot of such sections, we can actually determine the distance from this midline to the serotonin 2A receptor. And then we know that the layer 5, the layer we are interested in, is around 500 micrometer away from the midline. And this is very nice to know this, because then we can directly record from this area. And this is what we did here. This is basically glutamate release in the preliminary cortex, so the frontal cortex, which is a central region of psychedelic activity. And when we find, if we apply a psychedelic, this is what happens here. The glutamate release is desynchronized. So this is something that could not have been shown with any other methods, could not have been shown with EEG, and this parallels what people know from EEG. What is unknown so far, so this is unknown, of course, but what is also unknown, what subcortical areas do in parallel to this. And we see very often people believe that psychedelics primarily target the cortex, but it's also possible that they actually target certain regions in the subcortical area, and this might then actually have bottom-up effects. So as you can see here, if we apply our psychedelic, the effect here in the subcortical area is much more drastic than it is in the cortex. And this way we can actually expand on the knowledge. So if human research tells us, okay, the cortex desynchronizes, we can actually ask which cells are involved, what neurotransmitters are involved, and yeah, how is the subcortical region perhaps causatively linked to this. But this is ongoing research, and I hope we will publish soon so you can learn more about it. Okay, this was it. I have 9 slides. I think I'm already slightly out of time, but I think we will be fine. Just interrupt me, George. I have 9 slides remaining. We have now spoken about pharmacological principles, pharmacokinetics, pharmacodynamics, and we now want to use some of them to actually deal with certain findings in the literature and see what we can think of them. One hot topic is indeed psychedelic microdosing. I guess you have heard of it as well. Here's actually a Google trend analysis, which I did yesterday. As you can see from a certain time point here around 2016, there was a massive increase in people looking for microdosing. And this most likely has something to do with the popularization of microdosing by Fadiman, but also by certain articles which reported that microdosing is done in the Silicon Valley. Microdosing means that you every other day or every third day take subthreshold doses of LSD. This would be 5 microgram. Above it, you would actually feel it. So it's rather about those doses which you don't feel. And the idea is that this enhances cognitive functions, boosts physical energy, and promote emotional balance. The problem is very often in the studies that were done, they also use higher doses, which basically unblinds the people because they recognize what they were given. And in a study we performed at the Imperial College, with Laura Kärntner actually being spearheading the project, we actually found that, of course, if you have positive expectations at the beginning, when you start with microdosing, those people who have positive expectation will also be the one that later have large improvements in well-being. So this actually shows us that placebo does play a role. So it's important that when judging microdosing and the literature to go into studies where the rigor was sufficient, so double-blinded and placebo-controlled. If you're interested in the latest results, have a look into this review from Murphy et al. But I don't want to go too much into the question what the literature says, but rather ask a pharmacological question, namely, is it actually possible if we consume such a small dose of LSD? Is it actually possible that enough concentration would end up in the brain so that the receptor can be activated? And this can be approximated by going into a study from Holz et al. From 2021. Here they actually gave different microdoses and again did this plasma curve. So they basically determined, okay, if I consume 20 microgram, what is the highest plasma value? And they also did it for 5 microgram. And these values are usually given in picogram per milliliter, but you can actually also convert them into nanomolar. I did this here. I hope I didn't make any conversion mistake. Feel free to double check it and let me know. But if you convert these values into nanomolar, and then there's also another equation which allows us to, based on these values, predict the receptor occupancy. And then we can actually see with these different microdoses, 10%, 80%, or 28% of the serotonin 2A receptors in the brain are likely to be occupied. This is not a lot indeed, but it is enough so that we can rule out, okay, it's not impossible that microdosing does have an effect because there's a certain amount of occupation. So this is something you can basically use by knowing pharmacology well. You can make such predictions and then come for yourself to a decision. Does this make sense or not? But perhaps even more interesting than this association is the next question. Here's the receptor profile of LSD, here done by Ray in the year 2010. And these are also affinities. In this case, the higher the bar chart, the higher the affinity. And we see here's the serotonin 2A receptor. LSD actually prefers a lot of other receptors before the serotonin 2A receptor. And if we have a microdose that fills about 80% of our serotonin 2A receptor, this means these other receptors are occupied to a much higher degree. That means you microdose the serotonin 2A receptor, but you might macrodose these other receptors here. And this is important to have in mind because these other receptors are not linked to the psychedelic effect, and they can be found all over the body. They are involved in circadian rhythms. They are involved in cardiovascular system and temperature regulation. So there are all parameters which might be affected, especially on the long run, if you microdose. At least this is something we could predict based on these pharmacological considerations. So this is something important to have in mind. The next topic, so I have 3 topics for you. The next topic is DMT. As you might know, DMT is an endogenous drug, which means we can actually find it in the human body. Strassman, who dealt a lot with psychedelics, actually came up with the idea that the pineal gland and the DMT that is synthesized in the pineal gland might be important for near-death experiences. So this was popularized by Strassman, and a lot of follow-up studies were done to actually find indeed in blood and in cerebrospinal fluid and also in the urine, you find DMT. So all of us seem to produce DMT to a certain degree, and it is now widely accepted that DMT is a trace amine, which means we do produce it. The question is, however, does this make sense? Is it possible that DMT is produced to a level that it could induce a near-death experience? This is basically the idea here. Again, like with the microdosing, it's not about saying yes or no. It's rather thinking about it. And there's an interesting study by Dean et al., where they actually measured DMT concentrations at baseline and then after cardiac arrest. So they would basically euthanize the animals and compare the DMT levels before and afterwards. And they indeed found that after cardiac arrest, the DMT concentration is increased. So you might say, okay, this is in line with the near-death hypothesis. But if you come up with some pharmacological considerations, you might come to a different conclusions. Namely, we know if you go into the KI database that the affinity of the serotonin of DMT for the serotonin 2A receptor is 131 nanomolar in humans. In animals, in rats, it's 42 nanomolar. But as you can see, this is well beyond the concentration that was measured here. So we can actually infer that even under these cardiac arrest conditions, it's very unlikely that DMT was actually able to activate the serotonin 2A receptor to induce a near-death experience. Also, there's actually a very nice study by Gallimore and Strassman where they did pharmacokinetic modeling based on human trials where they injected DMT. And they actually, based on their modeling, said you need around 60 nanogram per milliliter in the plasma for a DMT effect to kick in. Again, this can be converted to nanomolar, and the results is 197, which is very close to the affinity in humans. So this seems to make sense. But again, in order to have a DMT experience, the concentration must be much higher than what we found in this animal study. So the result is rather against this hypothesis. This has also been critically discussed by Nichols, who is a very famous and big mind, a giant in the field. He also gave an interview to Alios Research, which is also very recommendable. So if you're interested in more arguments about for or against this DMT hypothesis, you can have a look in this one. And the last thing, and I'm through with you, is the last hot topic I want to come up with is the question or the phenomenon that all serotonergic hallucinogens are serotonin 2A agonists, so they activate the receptor, but there are certain serotonin 2A agonists which are not psychedelic. And the one that mostly are referred to are lisurit and ergotamin. Lisurit is actually a therapeutic. This is lisurit here. It's very similar to LSD, as you can see, and it was used, I think not anymore, but it used to be used in the context of treatment of Parkinson. Ergotamin is, I think, still available, and it's used against migraine. This is ergotamin. It has, in addition to this ergoline structure, it also has a peptide up here, so very bulky molecule. The thing is now, both of these substances have a high affinity for the serotonin 2A receptor. So low nanomolar affinity means high affinity. So both have a very high affinity for the serotonin 2A receptor, but they do not seem to be psychedelic. And this is really something that is hunting the psychedelic field, and everyone is very curious why this is the case. Funnily, if you measure LSD and lisurit at the serotonin 2A receptor, how good they are to produce this IP3 downstream of 2A, we find that LSD and lisurit are quite similar. So they are similarly have a similar efficacy. And then we are standing here, hey, wow, is it then possible that lisurit is not active? Similarly, when you compare LSD and ergotamin with regard to calcium release, again, both are quite similar in their efficacy, which is paradox. Now, the hypothesis that was put forward by González Meso et al., and they also provided evidence for this, is that lisurit and LSD are actually able to recruit a certain pathway, the classical one we talked about before with the IP3. But LSD, in addition, is able to recruit another pathway, which is this one is GQ coupled, this is GI coupled, and this might work via this hetero receptor by interacting with it. And this is basically an idea. This is out there and which has created a lot of attention and speculations. But the thing is, a lot of other researchers also tried to find other cascades which might be involved in LSD's effect, but not in Lisurit's effect. But we are actually at a state where it is still unclear because a lot of these things have not been replicated, and there are also paradoxical findings. And what I would like to suggest here is, although this might be true, it's not that I'm saying that it's necessarily wrong, but perhaps there might be also alternative ideas why they are not psychedelic. And here are some ideas. When it comes to lyseride, for example, we know lyseride, when you consume lyseride in a dose, so when you give it in a Parkinson, you actually have to increase the dose over multiple days, over weeks, more and more you have to increase. You have to start with a very low dose and only across weeks increase your daily lyseride intake. And the reason for this is lyseride is highly emetic. If you consume a high lyseride concentration just now, you would vomit for the next 4 hours. This could be a reason why lyseride is actually not psychedelic, because if you acutely consume a high dose and you vomit it out, then of course it cannot be psychedelic. On the other hand, if you do like the Parkinson patients did, you increase the lyseride concentrations over days, then it's very likely that you actually induce tolerance. We know for LSD, and we have reviewed it in this article here, when you increase the LSD doses over the days, you would actually prevent LSD from becoming psychedelic because tolerance develops. So it's possible that lyseride is not psychedelic because acutely it induces emesis, and when you increase it over the days, you get yourself into tolerance. This could be an alternative interpretation, and you might now say, this is very far-stretched. And I would like to give you another example, which is quipazine. Quipazine was long thought to be a non-psychedelic serotonin 2A agonist. The problem with quipazine is it also activates the serotonin 3 receptor, and again, this receptor induces vomiting. And Shulgin actually reported then in 2011 that if you combine quipazine with a serotonin 3 antagonist, preventing vomiting and nausea, that quipazine is actually psychedelic. So it's possible that similar things hold true for lyseride. When it comes to, this is my last slide, I think. When it comes to ergotamine, it's also a problematic compound because ergotamine itself is a rather toxic compound. It has a very limited therapeutic index, and if you overdose with it, you get serious side effects such as arterial vasospasm, and ischemic insults in your limbs. So this is similar to ergotism, which has been observed in the medieval times. So it's a rather toxic compound. It's not recommended to overdose. And then, although the literature is paradox about it, it seems that ergotamine has problems with actually going through the blood-brain barrier. The lower this value, this Kpuu value, the more difficult it is for a drug to go through the blood-brain barrier. And as you can see, it's very close to zero, actually. So it seems ergotamine does not really travel this barrier well. And even if it does travel, it seems that some of it might be kicked out by a certain glycoprotein, which is actually meant to kick drugs out of the brain. There are also 1 or 2 studies which show that ergotamine might go through the blood-brain barrier, but if you look into, or if you take everything together, it seems at least that it's not so easy. So it's toxic. You cannot increase the dose, number 1, and number 2, it might not be so easy to actually go into the brain. So this could be an alternative hypothesis for ergotamine not being psychedelic. The taking home message of all of this, of everything I showed you today is, and I hope this is what I got across, if you know a little bit the principles of pharmacology, then you can actually think for yourself what a drug does, where it goes to, where it binds, how long, with what kind of interactions might occur. And on these 3 hot topics that I mentioned to you, it's not really so much about saying it is like this or it's like that. It's rather about trying to critically think of the literature and not just take things as they are. And if you have doubts in findings, then this is actually a very good starting point for your scientific career, because you have yourself a hypothesis and can test whether your idea might be better than mine or the idea of someone else. Good. That's it from my side. I guess we will have a discussion now, perhaps. If not, these are yeah, sites where you can get further information. Thanks for your attention.